So does everybody have the files that you got by the Google Drive link downloaded? Okay, so like Dr. Murray said, they should all be like on a folder somewhere. Probably the desktop is simplest. And I guess I should get started. Are we online? Okay. My name's MarinWolf. I guess most of you were probably in the symposium. Yes. I need to speak up. Okay, I can talk loud, no problem. All right, so I have sent you all a PowerPoint, and I'm going to go over that piece by piece, and then we'll do some of the things I talk about in R. I kind of figured that most of the time we would be spending in R. My goal is basically to give you a toolkit and a basic understanding of the statistical models we use in genomic selection to relate genotype to phenotype using markers. So I'm going to we'll see how far we get. I don't know sort of the range of experience with R or with any of with statistics and that sort of thing. So hopefully what you'll have from this is a resource at the very least that sort of shows you a toolkit of R packages for doing genomic prediction. I'm here today. I'm going to do probably most of the talking up here, but Dunia is also here with me, and so when we're working in R, you can ask both of us any questions or problems that you're having, interesting error messages you may or may not get, that sort of thing. Okay, so I'm just going to talk for maybe 15 minutes or something to kind of go over some of the models that we're going to talk about, okay? So there are two classes of models that encompass how we make genomic predictions using markers. So the first one is using markers as predictors in kind of a regression type approach. So we're estimating the effects of markers, and then we'll use those effects and genotypes that we've taken on individuals to make predictions. And then the other kind uses the markers to estimate the pairwise genetic relationships in a population, and then based on having phenotypes on at least some of those individuals, you can make predictions about the genetic values of the unphenotyped individuals. So this is just a little to orient you. So marker effects based or marker assisted selection type approaches, well actually all of these approaches, since we're doing breeding here, we're interested in the additive genetic value of individuals. One of the key things to remember, so we're working usually with SNP markers that have 2 alleles at each locus. And what we're going to do is we're going to count the doses of one of those 2 alleles. I just want to make sure this is a familiar form to everyone. The idea that you'll have one homozygote coded as a 0, sometimes it's a -1, heterozygotes as a 1 or a 0, and then the alternate homozygote as a 2. So we're counting, in this sense, in this little graphic I have, we're just counting the number of big A alleles. And so if you do a regression of the phenotype on this marker dosage, the estimated slope is what we call the marker effect, and that's the sort of additive effect of that marker. And so what we're going to do in the GS models that are actually, at least that are marker effects based, we're going to basically estimate the effects of each of these markers and sum them up to get a prediction of the additive genetic merit of individuals. So in a situation where you have, let's say, marker-assisted or marker-based selection, where you have a lot fewer, you're going to have a lot fewer markers than you have individuals. And let's say you have one marker that tags each of a few QTL. This is a pretty simple model that you can fit using a multiple regression approach. So that's a fixed effects model. And I just put these 2 notations up. They're equivalent models. The idea is you have K markers. The marker scores for each individual are in this vector X, and then you have an effect for each marker. So if you sum them up, you get the total prediction, and you can write that similarly in this matrix form that you'll see a lot in the genomic selection literature. These are equivalent. So this is X here is the marker matrix. So you're going to have, and you're going to see this in the SNPs data set that I shared with you. Individuals are on the rows, and then their scores on, in this example, 4 markers coded 0, 1, 2 are on the columns. This is just another view of that matrix form for your reference. Okay, so in genomic selection, we're going to use dense genome marker data so that the idea is that we're not going to try to discover specific loci that are important, because we know for a fact that we can't really discover each one. We're going to discover the low-hanging fruit, the ones that have a very strong effect. So instead, we're going to genotype very densely across the genome and hope and rely on the fact that if we're dense enough in our coverage, each causal polymorphism that we don't know is going to be tagged, and that term tag means we're going to have one of our SNP markers, at least one, in strong linkage disequilibrium with each of the causal polymorphisms. So no more significance testing. Instead, so now we're going to have a problem, a statistical problem, where we have a lot more predictors that we're trying to use than we have observations. So one way to think about that problem is we don't have enough degrees of freedom to estimate properly the effects of each of the markers. And I want to try to illustrate another way to consider that. So this is going to do what we call, this is going to overfit the model if we just use our multiple regression approach with, let's say, 1,000 markers and 100 individuals. So this little example I have shows sort of a data set where you have some quadratic relationship between 2 variables. And so in one case, you could try to fit a linear regression through this data. So that's a statistical model with 2 parameters. That is, one is an intercept you have to estimate, and the other is a slope. But a slope and an intercept gives you a straight line. That doesn't fit the data well. So that means that this model here on the left is too simple. In contrast, so what we need to do here is add one parameter to give a curvature to the surface, and that would fit the data well. But actually, if we used all of our markers, all of our predictors, you would end up, or could end up with something that fit the data perfectly, right? So in the rightmost thing, the line goes through every single data point. So what that's doing is capturing both the signal and the noise in the data set, and that's not a good thing, because that means we're capturing environmental variation and all kinds of things. We want to get the balance between, we want to capture the genetic signal in our data so that we can make good predictions, because we have a certain data set that we're using to estimate these effects. We're then going to get new individuals that are genotyped but not phenotyped, so they're effectively new data, and we're going to try to predict their genetic value. So this model, this overfit model, would not be good. The solution that we're going to use is called ridge regression, RR-BLUP. You'll see a lot. It's also called, and there's a couple of aliases, so I'll call it random regression. It's also called SNP-BLUP. So this is a similar model to what we've already talked about, just multiple regression. But instead of a fixed effects model, it's a mixed or random effects model. So this is the matrix algebra form for the fixed effects estimate of the slopes, the regression slopes in a multiple regression, where X in this case would be your marker matrix. So with ridge regression, we just add this one new term, lambda, and lambda is what we call a shrinkage parameter. So it's defined as the ratio of the error variance to sigma squared beta. That is the variance in marker effects. So I'm going to try to give you an intuition of what this is doing, but the idea is that if we let each marker explain as much variance as it would in a fixed effects, we would have this overfit model. So instead we have this term that shrinks the effects equally across all the markers to confine the variance that the markers explain to something more reasonable. So another way we say this is that we're assuming that the marker effects are drawn from some universe of possible marker effects that is defined by a normal distribution. So we say the marker effects come from a normal distribution that have a mean of 0, that is on average the marker effects are 0, and that distribution has a variance, and that's what we estimate, sigma squared beta. So I want to try to clarify that and introduce you to the mixed model form that you'll see a lot in the literature, which is what I've written up here. So in this form, we have beta, which is your vector of fixed effects. You'll see this term a lot. Best linear unbiased estimator, or BLUE, is the statistician's term for the estimated or the solutions to the fixed effects. So fixed effects in this case are going to be things like location effects, reps, maybe blocks, some things like that. And then you'll see the term BLUP to refer to the solutions to, or the estimated levels of G, which in this case are going to be your marker effects. So this is where the markers come in. They're called BLUPs, best linear unbiased predictors. You don't have to worry at the moment about exactly what that means. And as I said before, we're assuming that these effects, so we're adding an assumption about, that we're not adding to the fixed effects, about these marker effects, that they come from this normal distribution. On average, the effects are 0. On average, also, the effects explain this amount of variance, this sigma squared. I wrote G here. It should be a beta. My main goal here is really to try to get you guys working in R. So what I'm going to try to go over now, and then in a moment we'll get into actually looking at these things, is what is actually in this model. So often we're going to write in matrix algebra the dimensions of the parts of the matrices and vectors that are in here as rows times columns. I just want to make sure everybody knows how to read this. So of course y is just your response variable. It's just a vector. You have the fixed effects term, which has what we call an incidence or a model matrix, and I'm going to show you what's inside of those in a moment. Those are going to relate the phenotypic observations in your data to the levels of the fixed effects. Okay, so it's going to be a matrix that's going to be the number of rows are going to be the number of observations. The number of columns are going to be the number of levels of fixed effects. So like if you have 3 locations, you should have 3 columns, or actually 4 plus an intercept. And then beta will be a column vector with the number of rows corresponding to the number of levels of fixed effects. Error variance is also a random variable, mean of 0 and a certain variance, sigma squared E. And then, of course, so in ridge regression, BLUP, Z is now your marker matrix. So this relates phenotypic observations, marker scores for each individual. So these are just your predictors. G is now going to be a vector of the BLUPs that correspond to the marker effects. And again, we are making this assumption about, you can think about this as making an assumption also about the genetic architecture, that on average every marker has an effect. It's very small. On average it's 0. So this corresponds, I'm sorry, to the infinitesimal model. Okay, so I want to show you the incidence matrix, because to use some of these R packages, we have to know how to create them. It's actually pretty easy to do in R, but you have to know what they're supposed to look like. And I feel like it's very helpful to do that, because when you understand them, then you're going to have a lot less headaches. You're going to have a lot less complaints from whatever R package you're trying to use. So this is an example of an incidence matrix where you have an intercept. So this is, you're going to have one row for every observation. Everybody gets a 1 in the first column because everybody has the same intercept value. And then in this example, 3 locations. So that what we do then is we have a score of 1 if in the column where the observation was made. So these first 3 rows were made in location 1 or location A, so they get a 1, and then 0 elsewhere. So this is just telling the model where the observations were made. And you can include continuous covariates, and they appear, and this is important when you think about the incidence matrix we use with the markers, because they are continuous covariates, predictors, and so those go in as a single column with the actual values of the predictor. Okay, so so in this case for fixed effects, it could be something like planting date, I don't know, flowering date, temperature, depends on your experiment, but that's what I mean. For the random effects in ridge regression, those continuous predictors are the marker dosages. It is your marker matrix. Make sense? Okay, so let's jump into R. So the first couple of things, I don't know everybody's experience level, but we get data loaded, and we can do ridge regression pretty quickly. So everybody has this loaded. Okay, so these first couple of lines you can just run, but all we're doing, so firstly, I think you guys must have covered working directory. You basically have to tell R where the data are located. And so you should have downloaded a folder. This is just the location on my computer. And so if I run these 2 lines of code, we're just loading 2 data sets. And then quickly subsetting, I just wanted to delete one of the years. So I've given you an example data set. This is actually cassava training population from Uganda. So what you'll find is there are 3 locations. In each location there are 2 replicates, and there are about 411 individuals, I believe, in this training population. So if you're using RStudio, you can actually just click on this to look at the data set. Just so. Okay. Yes. Mm-hmm. Okay, you're up and running. Yeah, so you should actually, you could just click download the whole folder. Well, that's fine too. No, that's fine, yeah. And then did you also download the R code? Yes. Okay.
Put it in the folder. Where is that one?
So that's this.
This one. And I'm putting folder, you know. Is it there?
Maybe just leave it on the desktop. Would be simple.
Okay. So this is 1 second, then 3.
Yeah. So this should be the R.
So now I'll upload.
So this should be the R script. So we might be able to.
Oh, the view is probably. That's where you can't think of it.
This computer doesn't want to do it. Okay, you can also just click open here, and then desktop. Okay, where is the R script? You see it? Why doesn't it show it? Do you see?
What do you mean?
I'm looking for the file.
But which file?
There's one in that folder, so you might as well use that folder.
That's in the folder.
Can you? Or you might actually right-click on it and say Open with R Studio.
I have opened, this is R Studio, This is R Studio.
Okay, so no, go to the folder where you saved the file, the R script.
File.
Okay, yeah.
Yeah, I got it there too. Yeah, you see very close. You click on it, and then you can actually look at the data.
It is not done. It is not.
Or you can actually build it.
This is my file. This is.
Mm-hmm.
So go on desktop.
Yes.
Okay, and then
Yeah, but it's a problem. Popped out.
Huh.
Showing demonstration.
there's no folder that's showing, right?
Okay, here's another way. So cancel this and actually just open this folder.
Okay.
Not in R. Yeah, and then right-click this. There's no open with. Or maybe click open.
Okay.
And then, okay, now it's going to ask you what software to use. And so now you can just find R Studio, hopefully. Uh-huh.
What? I should delete this one.
Wait, okay, so hit browse here.
Oh, yes.
And then go to desktop. And then,
yeah.
oh, we're actually looking for the software. We're trying to tell it what software to use to open, and it's not finding R Studio.
I can delete them again. I can download it.
No, so the file isn't the problem. It's the software. Where's R?
R is there, So R is there. R is this one. I've downloaded. No.
So this is the actual data.
This is. Oh. Then where is R? This is the file.
Wait, so you have R open, which means...
This is your file, I've downloaded this. Oh.
Yes. Okay, click this.
And it is.
I don't think that's the problem.
To load it from the desktop.
This is your folder, right? By just tapping for the
This is working good, right?
Right. Some people need to go, and I never know this.
R Studio doesn't want to open things?
doesn't show here.
Like, everybody is? Oh,
You're also
okay. This could... Okay. Do you want to see if you can help him out?
Yeah, what do I need?
We're trying to get the R to open.
Vernon, are these steps supposed to be in, like 0.69 in like a frequency?
Aha. Yeah, I should tell everyone.
Okay, I just want to make
Yeah.
sure it wasn't the wrong data.
No, it is. It's they've been imputed. So imputation, the software we use to do that gives probabilities
Probabilities, got it.
because it's based on regression. So 0.06 would be like a score of 0, very close to.
Very close to 1. Which should be a heterozygote.
Yes, you got it.
Could you make the console larger so I could see what commands you have?
Sure.
Nolan, you can't make the console better hands on there.
Can we
Like the maximize?
make this bigger? Okay, so I think I have the impression almost everybody has the data loaded now. So I just want to show you the data, and I will show you my console So all I did so far was run the first few lines of code. So that's here. So set my working directory, loaded the phenotype data set, loaded the SNP data set, and I also ran this one line here, which is just making a copy of the phenotype data set, but I've subsetted it using this subset function to only the year 2013. I chose to do that just because it's a little simpler. So in this data set, we have 2 years. In the first year, the trial was done in 1 location. In the second year, it was expanded to a 3-location trial with 2 replicates in each location. Okay? So this is what, and so these you should see in your R script. These are the first 4 lines. This is just loading the data. There's also some comments up here at the top that just kind of tell you what the data set is.
Martin, so are you recommending that we click on the file in our folder that's the script?
Yes.
Upload that?
Yeah, so that'll provide some example code, and you guys are free, should feel free as we go along to kind of mess with it. But I've put it together in such a way that you can just run it line by line without any modification, and it should give you an example and a little bit of explanation of every analysis that I want to show. So I think most of you probably find, I'm guessing that most of you find that the phenotype type data set is pretty straightforward. You know, so we have just, I've provided 4 traits and then some design variables that we'll need to account for. The SNP data set, I sort of made an example of what those look like normally in my little presentation. So we have individuals on the rows, and so this is, so you can't actually see it here, but there are row names that correspond to the individual's identity, which is coded 1001 through some other, so they're just coded numbers, and they should match the variable clone in the phenotype data set. And then you have these SNP codes here. So these are just the names of the markers along the column names. And so those are coded S1, which corresponds to chromosome 1. This isn't actually important for the analyses we want to do, but for your information, I've given you 1,000 markers that are distributed approximately equally across the genome. So this would be chromosome 1, and this is the base pair position in the chromosome. And then the codes are a little bit different than what you might expect. So most of them are 0, 1s, and 2s, like I described. You might notice some other values, like 1.99. And so what happens is, in my data sets, we're using genotyping by sequencing, so we get a lot of missing data. And so we have to do imputation to fill in those missing values. And imputation will give you some variation along the same scale. So they'll be between 0 and 2. But so 1.99 means we're pretty darn sure that this is a homozygote. You might see something like 1.5 or 1.1. So don't worry about that. It doesn't affect the analysis because, again, we're going to do these things that are basically like regressions. So as long as these are continuous and along the scale that we expect, we're fine. If you see something that's more than 2, let me know, because that would be really weird.
How would this format handle that?
Jeff? 1 through 4, maybe?
Kind of practice the work with tetraploid data, but we've also developed a tetraploid data version that's being reviewed right now. So it wouldn't be from 0 to 4. That would cause error. Well, that would be that's the right way to parameterize it. That doesn't allow that. You have to basically
Oh. So my examples for a diploid,